Okay, so getting up and running here. So we are talking about DNA. So this is going to kind of revisit us back to previously, where we learned about like mitosis and meiosis. We learned about why it's important that DNA is accurately represented and accurately copied. But how does that work? How does it copy it? That's kind of one of the main focuses of this lesson. And what is your DNA form? So has anyone ever seen this picture? Does anyone know what this is or think they know what it is? So this photo here was taken in 1951, and this ended up winning a Nobel Prize. So this is our photo of DNA. It's the first ever photo of DNA. They managed to isolate it, and they managed to take a picture of it. And this kind of revolutionized science from this point onwards. It doesn't look that impressive, but it's quite monumental. Because it confirmed a couple of things. The big thing it confirmed is that DNA is, whenever we've done like pictures, we've done it like this, where it wraps around itself. So DNA is kind of like a corkscrew here. It winds itself together. It goes around and around. And this confirmed that there is, in fact, a double helix there. This confirmed that there is, in fact, a double helix here is what we call this. It confirms that DNA is attached. We theorized what it looked like, but this was actual confirmation of it. So we're going to learn what all this stuff means here. There's a lot of stuff with this. We're going to learn what it all means here as we kind of go through this. Let me just delete my. Okay, so starting out here. So, oh, sorry, this is chapter 12, by the way. This is chapter 12. So we're looking at DNA. What the heck is DNA? Let's see. There's going to be a lot of the stuff we don't need. You don't need this. None of this is fine. But again, so after we looked at Mendel's work, we began to look for what actually is involved in inheritance. We know that inheritance is a thing we can control. There's a mechanism in our body that does it, that passes on genes. They wanted to try and figure out what it was. And so what they did was they tried to figure out if it's DNA or if it's a protein. Which one of these two things is the thing that does it? They didn't know. Same thing, you don't need this. So we're kind of going through a little bit of a history here. The first one we have Griffith. So what they did was they started looking for this material. So they ended up injecting, I think that's pneumonia is what that is. I'm trying to think of what the Latin term is. They injected mouses with stuff. They injected mouses with some type of a disease. And so this is what kind of set this stage. So basically what they did was they started injecting different proteins and DNAs, et cetera, into mouses, and they tried to see what's causing it to live or to die. And sometimes it's exposed to heat or not that important. You don't need to memorize this. We eventually ended up looking into a person named Avery. Again, don't need this, just a history lesson. They figured out that there's a molecule that transforms the R strain of this into an S strain. And we'll talk about what that is later. But basically, they figured out that when these S cells are killed, DNA gets released. And so we get people who just kept kind of taking this concept and going deeper. The next people we had were Hershey and Chase, and they started using radioactivity to label and track. And they figured out that DNA gets injected into the cell, and then that provides that information needed to produce new viruses, specifically with bacteria and viruses. So they looked at how do viruses spread, for example. How do they grow? How do they inherit stuff? This is kind of taking us to our big thing here. So this is where the stuff that we actually need is. What is DNA? A lot of what DNA is something called a nucleotide. This is a structure. I will enlarge this photo in a bit. But basically, nucleotides are we call them the subunit. They're these smaller parts of nucleic acids, which we'll learn about soon. And they have a couple different things. They have 5-carbon sugars, they have what's called a phosphate group, and a nitrogenous base. We will learn about all of these as we go through. This is something you do want done. So, so these things here are a bunch of examples of these nucleotides. So we have structures, we have purine bases, we have pyrimidine bases. Again, we will go over all of this. So we will go over what all of this is. Am I on the right file? Yes, I am. Here we go. Sorry, I need to wipe all my stuff here. There we go, delete that. Okay, so oh, come on. Here we go. So DNA structure, we don't even know the history behind it, but basically one guy eventually figured out some DNA structure stuff here. So he used something here, we call this Chargaff's data. So he looked at the amount of, we call them A, C, T, say A, G, T, and C. He looked at these things. He looked at these things, these nitrogenous bases in living things. He took a bunch of different species and he analyzed what makes up all of them. So we can see that, so bacteria here has 26, percent A's, but humans have 31%. We look at these C's, humans have almost 20%, but yeast has 17%. So he just looked at the structure of all these things. But one thing he kind of came up with was a little bit of a rule. He came up with a rule that, and this is a really important thing here. Whenever we have a C, we usually have a G. And whenever we have a T, we usually have an A. And this is what's going to make up the entire foundation of how you build DNA. You don't need the chart. Don't worry about that. So where he got this rule from, the C equals G and T equals A, is he started looking at these base compositions and he kind of noticed the pattern. If we're kind of looking here, if I look at the rat, the C and the G, those are linked. They're almost the exact same numbers, for example. If I look at the A and T of it, it's almost the exact same percentage. And this trend carries true. This one, not as much right here, but for the rest of them, it's pretty accurate. There's a couple of minor differences, but it's pretty consistent throughout. So this is where he got that from. So our search for the structure of DNA was led by 4 scientists. Rosalind Franklin is the most well-known here. This is the person who took that photo that I showed you guys, the actual photo. So they eventually looked, so where we went from this to, okay, let's actually find DNA. We think it's DNA. DNA is the thing that passes inheritance. These 4 people are going to start leading that. You don't need to work, you don't need to know. The only one you need to really know is this person, Rosalind. Don't worry about the other ones. But Rosalind's, I've seen questions on that one.
I think I did it. I just sent you a picture of both.
Okay.
Things.
I'll take a look here. That looks about right, yeah.
I got creative with the baby.
Okay. So how did they do this? So one thing they figured out was they used something called X-ray diffraction. We don't really need to know what that is. We just need to know that they used it and they found out that DNA is, we call it a double helix. If you want to, like, if you're trying to envision that, it's a ladder, but you just twist it. You just twist a ladder, basically, and it kind of curves. There's that picture we showed you at the start. So X-ray figures out that DNA is a double helix and that this is formed by 2 strands of nucleotides that are twisted around each other. Okay, so again, so that double helix, that twisted ladder. Again, that's what this is. It's a ladder that kind of got twisted up. And again, this is like, what a diagram of it would be. So I want to kind of zoom into this C equals G and A equals T thing. If you guys look, these are kind of color code, right? You're going to look, any red one you see, there's always a timing, a T, an orange one beside it. So they're always linked like that. So it's a rule where, anytime I have a D9 and A, I need to have a timing, a T. And then you see the other one, right, with the blue and the green. You never see an example of, for example, red and blue. It doesn't work that way. We don't need to worry about this. Okay, so we already have this. This is fine. Let me wipe all this ink. Oh, it's not letting me erase it. Sorry. Okay. So we don't need this. We know it's like a twisted ladder now. So the structure of DNA. We don't Yeah. Yeah, okay, you know what? So when you have the ladder, right, we think about a ladder. I'm gonna unwind it. You know, this is what it looks like. Those rails of it, the rails of it, that the steel that kind of holds it all together, those are, it's called deoxyribose and phosphate. And then those steps, just like we saw in that picture, are the cytosine guanine or thymine adenine form those steps. And again, that's just that C and G and T and A. Most people remember it just off the first letter of it, right? So just kind of visualize this. Again, when I'm like, it's only adding the 3' at the bottom. We will see a lot more visualizations of this. But again, as we're kind of going it, remember if we synthesize in the 5' to 3' direction, which means we are moving this way. So when we are adding on a new DNA strand, when we are combining DNA, that's the direction it goes. And so it slowly gets added on here and here and here. And for example, if this is an A, I would know this would need to be a T. If this is a C, it has to be the G, and so on and so forth. So that's how we're actually combining those two DNA strands. If you remember, this is the ladder, right? We'll later twist it up. We don't need to worry about that. So chromosome structure. So when we have prokaryotic cells, if you remember, those are the simple cells. It doesn't have a nucleus and stuff, right? So that those DNA molecules are in the cytoplasm, and that just has a ring of DNA and all the proteins and stuff. In the eukaryotic cells, though, that's when those DNA is organized into those actual chromosomes that we've learned about. Okay, so we're almost done. In fact, we are basically done. So those chromosome structures, and again, this is in eukaryotic cells. So these are eukaryotic chromosome structures. So DNA is vast, it's huge. So it needs to be able to fit places. We like compact stuff. So that's why it winds up for one. That's why it does a double helix. It's efficient. You store a lot more information in a smaller space. The second one is that DNA coils around of proteins called histones. So when you get DNA and you get histones, this is that nucleosome that we've been looking at for so long. Remember we've seen those diagrams in nucleosomes? That's what those are. So when we group these together, we get chromatin fibers, and then we coil those to form a chromosome. So there's a lot of efficiencies here. So DNA, we go from double helix, and then it goes to histones, then it goes to nucleosomes, then it goes again to chromatin fibers, and then it turns into chromosomes. So it's just winding itself up closer and closer and closer. It's grouping up to take as little space as humanly possible. So we'll end up doing section 2 today. This is just section 1. So what are we at? We're at 12:20 right now. So I'm going to give you guys till 2:15. You'll take your break at 2. We'll come back at 2:15, and then we'll start talking about 12.2. So what you guys can do is you can start reading the textbook there, because it's helpful to see some of those pictures and to get that extra information there. I'm going to start having a bunch of stuff that actually hand out for this one. So you guys can read that and complete the essentials. Now, there will also be a study guide for this that you'll do, because this has a lot of stuff where you kind of just need to put some pen and paper and work some stuff out. I will hand this out tomorrow. We don't need to worry about it today, but this will end up being handed out later. So you guys about 40 minutes there. Read 12.1. If you finish that, you can start reading 12.2. At 2:15, we're going to go over 12.2.